All right, people are starting to stream in. We're going to give it a moment while everyone gets on in today. So let me just start my slides for 1 second while we get everyone in. So hi everyone. For those of you who have not met me before, I am Lauren Traster. I am the 4-H Teen and Leadership Program Coordinator here at UVM Extension. So we always like to get started introducing ourselves. Hi, Simon and Jack and Emily. These are some of our regulars. So if you're new and what we like to do is have everyone find the chat box, go on in, introduce who you are and where you're from, because we, like KL's from Maryland, I was just about to say, we get mostly Vermonters, but we do get teens from all parts of the country, which is very cool. Hi, Sage, nice to see you. Well, I don't get to see you, but nice that you're here. So I recognize a lot of the names of people coming in today, which is great. So go on in, introduce yourselves. I'm going to pop into the chat box right now. If you need our live captioning services today, just click on the link that I just put in, and that will bring you to the live captioning where you can follow along with the presentation today. So as you all continue to introduce yourselves, and I'm going to encourage everyone to go on in and introduce yourselves, because it just makes it so much friendlier to know who's here and where you're from. But as you continue to introduce yourselves, let me just go over our protocols while we hang out here in Zoom land. So first of all, you are all muted, but that doesn't mean we are trying to silence you. We welcome and encourage you to share your thoughts and engage with our presenter today. And there's 2 ways that we do that. The first is through the chat box, which you're all using right now, which is great. Hi, Egan from Wisconsin. Glad you could join us. So we use the chat box to talk back and forth with the presenter. I know Matt's going to ask you a lot of questions today, and we'll be eliciting feedback from you. That's how we use the chat box. The Q&A box is to ask your questions to the presenter. So we keep those things separately because we may not get to your questions right when you ask them, but we do get to all the questions before we wrap up. So put your questions in the Q&A box, use the chat to talk back and forth with the presenter, and also just share thoughts on topic with one another. If you guys get too far off topic, I will kind of call you out for it. So Kat, I'm gonna introduce the topic in a moment, so just hang tight. So just make sure there's no distractions. Really, the only way you can just be distracting is if you use the chat box inappropriately. So just stay engaged, participate fully. Before I introduce today's presenter and the topic, I do want to mention a couple of upcoming opportunities that some of you might be interested in. So we just launched 2 4-H Teen Time programs. One is starting next week. It's called Designing Your Future, and it's a career exploration 3 workshop series. One next week, identifying your career interest, followed by showcasing your essential skills. And then the last is navigating your own career path. It's going to be every Wednesday for 3 weeks from 12 to 1. So it's a lunchtime. Most people are remote on Wednesdays. They will be recorded if for some reason you can't attend and want to. The other is Shaping the Future. We're offering 8 scholarships. Unfortunately, it's only for Vermont teens, but you need to be in grades 9 through 12. But we're offering full scholarships to attend the National 4-H Conference, which is going to be a virtual conference this year. And it's really cool because at that conference, they identify what they call roundtable topics, and you get put on a topic and you do research and you prepare what they call a briefing, and you actually present the information to people who work in those government agencies that work on that topic. So you can find out more information at the website that's listed, but if that interests you, go ahead and apply, because it is an application process. and you need to have your application in by February 10th. The last is in New England. The New England states are putting on a teen meet and greet, and that is tomorrow night from 7 to 8. And you can meet 4-H teens from all over New England. And I know they're going to be doing some activities, and it's just a way to sort of like gather with other teens. I'm not responsible for that program, so read the flyer, check it out, register if that interests you. There's 2 more opportunities. I just found out yesterday that our national Teen Science Cafe network has an opportunity tomorrow at 11 a.m. to hear from a particle physicist who works on the, sorry, I can't see my notes, who works on the Hadron Collider. Sounds really cool. So if physics is your thing and you're available tomorrow at 11, I would go to this website you see listed and sign up. I know space is limited. I'm not sure how many seats so they have, but check that one out. And then finally, our friends over at the Vermont Brain Club are starting their sessions back up. So next Tuesday, they're doing a session on the relevance of neuroscience in society. And so if you're interested in the brain and neuroscience, go check out what they have going on. So those are some opportunities for you all. And then let's just move into today's presentation, which is harnessing microbes to protect the environment. Our presenter today is Dr. Matthew Scarborough. And let me just tell you a little bit about Matt. So Dr. Matthew Scarborough is an assistant professor in the Department of Civil and Environmental Engineering at the University of Vermont. His research focuses on the application of microbiomes, also known as microbial communities, to protect the natural environment and public health. He currently focuses on management of waste from agricultural processes with the goal of harnessing microbiomes to convert renewable feedstocks to valuable products. So this work aims to provide additional revenue for farmers while reducing water and air pollution. His work encapsulates many scales, from studying the function of individual enzymes to implementing full-scale microbiome-based conversion platforms. So it is my pleasure to introduce Dr. Scarborough to you all. So let's give him a nice big welcome, and thank you for joining us today.
All right, well, hey, thanks for having me. I'm gonna try to share my screen, and hopefully you can see it.
Yep, we can see it.
Excellent. All right, and so thanks again, everybody, for coming. Lauren already introduced me, and as she said, I'm an assistant professor at the University of Vermont, and today I want to talk to all of you about microbes and how we use microbes to protect the environment. And so I'm an environmental engineer by training, and I worked as an environmental engineer for several years, designing and implementing different projects across the United States. And now I'm back at the University of Vermont teaching and doing research. And so there's 3 things I'll talk about today. So I want to start with just a brief overview of what microbes are and sort of what they do. And then I want to talk about how we can use microbes to clean water. So as humans, we take water from the natural environment, we use it in various ways, and then we want to put it back into the environment. And before we do that, we typically clean it. And then I also want to talk about how we can use microbes to produce renewable energy. And so I'm going to give you some examples of all of these, and I'm going to get your ideas about some of these. And I'm hoping what you take home from this today is that microbes can be a really, really useful tool to help us take care of the environment and to reduce human impacts on the environment. And so, what are microbes? Somebody just asked that question in the chat too. So that's where we're going to start. What are microbes? And so I want to ask all of you to get started. What are microbes? And you can answer this question in the chat either by giving me an example if you want, if you can think of any, or giving me a definition. So what is a microbe? And so I'm going to wait just a minute here and see, and all answers are welcome. And so please let me know what you think.
And even if you aren't sure, make
Yes.
an educated guess, because that's
What
how
is a
we
microbe?
learn. What do you think it might be? And there is a delay. So here we
Yeah.
go. So Jack says microbes are very small things that can sometimes make up larger things. Oh, sorry, that was Simon speaking. Thank you.
Yep.
Brooke says a microorganism like bacterium that causes disease. Kat says a microscopic thingy, and Anna says microscopic size organisms, including
Great.
single-celled organisms.
I like all of those answers. Extremely
And
small
KL says
creatures.
extremely small creatures that can't be seen by the human eye alone.
Excellent. All right. So here's what I consider microbes. So microorganisms are organisms, for the most part, living organisms that are too small to see with the naked eye. And so somebody almost had that definition exactly. And I wanted to give some examples because there's a wide variety of microorganisms, so much diversity across the tiny creatures that make up our planet. And so there's viruses, which we can argue about, and actually scientists do, whether they're living organisms or not, but they certainly play a major role in the microenvironment. We have bacteria and archaea, which you've probably heard of bacteria. We're going to talk about bacteria and archaea specifically today. And those are both unicellular, and somebody said that they can be single cellular organisms, and that those are the two examples. There's also multicellular organisms that can be microorganisms that we still can't see with the naked eye. And these include fungi, and I know you've all heard of some fungi, and some are microscopic. Algae, which you can sometimes see, protists, helminths, and rotifers. And so if we wanted to sort microorganisms sort of from smallest to biggest, this is what it looks like. And so you've heard a lot about viruses lately in the news. I'm sure you've all caught up on coronavirus, and here's that picture, a common illustration anyway of coronavirus. And so we have viruses, bacteria, fungi, protozoa, algae, rotifers, helminths, and protozoa. And what we can do as engineers is get these organisms to do some desired function. And oftentimes that's getting rid and cleaning up some pollutant that we don't want, or taking our waste material and converting it into something more valuable. And so the one key, with the exception of viruses, all of these organisms are living. And so as a living organism, microbes need certain things. So like humans, they rely on their body producing energy for their body to function. And so they move around, some move around, they have little flagella that they can use, and they spin and they can move throughout their environment. They need to produce more cells. Microbes are very interested in just growing and dividing and making more cells. And this energy that microbes rely on is comes from food in a lot of ways that humans sort of rely on food to get our energy. And so food for microbes can include a whole bunch of different, very small chemical compounds. And in a lot of cases, they eat some of the same things that we do. And so sugars, fats, proteins, if you look at a nutrition label, you see all of these things. And what microbes do with this food is they break it down and they harness the energy from this food. And many microbes that do this need to breathe. And when I say breathe, what I mean, it's that when they break down their food, here's a somewhat complicated figure, but I'll walk you through it. They take this sort of energy-rich raw food source, they break that down into some sort of energy poor byproduct. And through that process, they're able to capture useful energy for themselves to use. They also generate electrons. And I know you all have heard about electrons. And so they can use these electrons, and they do what we call dumping those electrons onto some terminal electron acceptor. And through this process, they're able to generate more energy. And so as an example, if we think about what humans are doing, we take energy-rich food, we break that down into CO2, which we exhale. And then not only do we get energy from the food directly, but we take electrons and we pass those on to oxygen, which oxygen we need. It's what we breathe in through our lungs. And we use this to accept electrons, and we make water in this process. And that allows us to produce even more energy. And so there are microbes that do just this. They break down energy-rich food into energy-poor byproducts, and then they'll use oxygen as what we call a terminal electron acceptor to generate more energy. And so knowing that microbes need energy sources and need some sort of terminal electron acceptor, we can start to engineer conditions where microbes can sort of be forced into doing something that we want them to do. And so the first example I want to talk about is using microbes to clean our water. And so I have another question for all of you, and you can just type it into the chat, but I'll give you a few options. And so my question is, if you know, where does your wastewater go in your house when you're done with it? And so flushing the toilet, letting water go down the drain, where does that water go? And I'll give you three options. One, a wastewater treatment plant. Somebody said the sewer. So if it goes to a sewer, you can put that too. It goes to a sewer and then a wastewater treatment plant. The other option would be a septic tank. So maybe you live in a rural area and you have a septic tank. And the third option could just be, I don't know. And that's totally fine. Septic tanks.
I bet we're going to get a lot of septic tanks.
That's how I grew up. A and B depending. I don't know.
And I'm not surprised at when I was your age, I don't think I ever thought about where my wastewater went. So I'm impressed that many of you do know. I think when you are rural, you probably hear your parents talking a lot about the septic tank and needing to get it, cleaned
Pumped
down every
out.
7 years.
I was one of those kids. The truck would come and empty out the septic tank.
Yep.
Cool. So in the United States, about half of the population's wastewater goes to a septic tank, and about half of the water goes directly to a wastewater treatment plant. One of the kickers, though, is if you have a septic tank, when that truck comes, they'll pump out the wastewater, and it usually ends up at a wastewater treatment plant. And so it could be indirectly, but a lot of your water, after you use it, is going to end up at a wastewater treatment plant. And so since a lot of you do have septic tanks, I will say, though, when we send our water into a septic tank, it's being broken down by microbes. And I'll come back to sort of how septic tanks work at the end of this talk. But I first want to talk about how at a wastewater treatment plant, we can engineer processes to make our waste, to clean our waste before we would send it back to the environment. Oh, there we go. All right, so why do we want to clean wastewater? And so there's many reasons, but I'm sure you all have seen polluted water. A lot of times, what we contribute as wastewater, if it were left untreated and returned to the environment, we'd see a lot of a lot of potential water quality issues. And so one thing that can happen is if you send a lot of organic waste into a stream, the microbes in the stream will actually consume that waste while consuming oxygen. And they consume all the oxygen so that fish no longer have enough what they need to live. And so it can result in large fish kills and killing off of aquatic life. And the other big issue we see, if you've ever been to Lake Champlain in the summer, for instance, the algae blooms that are occurring in Lake Champlain are largely a result of nutrients entering the lake, so nitrogen and phosphorus, for instance. And so we've been using microbes to help us treat wastewater for a long time. And so this is a picture, one of my favorite pictures, of a wastewater treatment plant being constructed in the early 1900s. Oops. And so these, what you see here, this is called a trickling filter. And what we have is rocks, essentially, in a tank. And they would put wastewater over these rocks. And they knew that it could be cleaned up and that it would remove the ammonia in the wastewater. It would remove the organic carbon in the wastewater. And through this process, even though they didn't know it was microbes doing it at the time, they could clean water significantly. And so what was happening, it turns out, is microbes would grow on these rocks. And as the wastewater passed over the rocks, the microbes would consume the organic matter, cleaning it up, and they would also remove the nutrients that we want to get rid of. And so the way that we've sort of engineered this process now is something, it has a great name. It's called the activated sludge process. And so in this process, we have what you can see is just a brown sort of muck, for lack of a better word. And then you can also see this white stuff on top. And so what this is actually it's oxygen. So we provide air into the system at the bottom of the tank, and you're seeing little bubbles forming on the top. And so the microbes in here, some of them are going to consume the waste organic matter that's in wastewater by also consuming the oxygen. So in a lot of ways, these microbes are acting as we do. They're breathing with oxygen. And their food in this case, though it sounds gross, is the organic matter that's in wastewater. And so this is what a schematic of this process would look like. And the key here is that we have 3 different zones. Typically, we have an anaerobic zone. And by anaerobic, I mean that there's absolutely no oxygen present, that there's no other sort of external electron acceptor or terminal electron acceptor present. And then we have an anoxic zone where there's no oxygen, but there's other terminal electron acceptors present. And then we have an aerated zone where there's lots of oxygen present. And so if we do this process, what happens is we can grow microbes through this process, and then they can settle out in a second tank, something that we call a clarifier. And so if we zoom in on each of these zones, there's several different things that can happen. And so I want to actually start with this zone 3, this aerobic zone that we use. And so in this zone, we have several different types of organisms. We have bacteria that are converting organic waste to CO2, and we call these, a big word for you, ordinary heterotrophic organisms. And then we also have bacteria that are removing phosphorus. And phosphorus is one of these nutrients that, if left in the water, can cause algal blooms. And then we also have organisms that are converting ammonia to nitrate. And so there's actually two different classes of organisms. One we call ammonia oxidizing bacteria and the other nitrite oxidizing bacteria. And if we go back to an anoxic zone, we have organisms that are actually going to produce and convert the nitrogen in the system into nitrogen gas, so that we have the organic carbon leaving as CO2, and we have the nitrogen, the ammonia, ultimately being converted to nitrogen gas. And then in this anaerobic zone, we have these phosphate accumulating organisms that accumulate phosphorus in the aerobic zone. But the first thing we do is we stress them out. We stress out these organisms. And what we've found is that when we send these organisms then into an aerobic zone, they're going to uptake a whole bunch of phosphorus. And so we can play with these different zones to get microbes to do what we want them to do. And so in an activated sludge process that I just showed you, we have microbes that are converting organic matter to CO2. We have microbes converting ammonia to nitrogen gas, and then we have microbes that are taking up all the phosphorus. And in this process, they're generating new cells. So these microbes are growing and dividing. And when we send these microbes into a tank, they can settle out, and then we produce what we call sludge. So what we're left with at the top of these clarifiers is relatively clean wastewater that we're going to take, we're going to disinfect, and then we can return to the environment. And so, are there any questions about wastewater treatment before we move on to another use of microbes, which is making energy? And I'm sorry, my screen keeps moving around here. Let me try this. Okay.
Matt, there are
Yeah.
a couple of questions in the Q&A.
Okay.
So if you want, we can just address those right now.
Yeah, let's do it.
All right, so KL asks, what does it mean to be capsid-based?
Oh, yeah, sure. And so capsid-based, so specifically, we're talking about viruses. And so they're not living cells in the typical sense of a cell. They have protein encasements around them and sometimes lipids that are around them. But we call this sort of bit that encapsulates the viral DNA or the viral RNA a capsid. And so they're not really cells per se. And that's what it means to be capsid-based. Does that help?
Two related questions. So what is the least complex organism there is, and what is the most complex organism there is?
Well, let's see, the least complex organism. They're all, as it turns out, pretty complex, but I study bacteria and archaea. And so the bacteria that are least complex have about 150 genes, something like that, compared to the millions of genes that humans have. The kicker is, though, as scientists, we're still trying to build a minimum genome. So we know that with about 76 genes, I think, we can create an organism that will grow and self-replicate. The kicker, though, is that we don't know what every single one of those genes does yet. So even microbes that have a very small genome and this really constrained ability to take DNA and make RNA and then convert that RNA into proteins, even the simplest ones we don't fully understand. So it's hard to even say that they're not complex. The most complex organism, I mean, I still find humans perplexing, but octopuses too. So I don't know, I would go with those. But at the microbe level, we have microbes that have, again, several million genes in their genome, and they're quite complicated. But also, we're starting to find out that there's one of the more interesting, really complicated ones are what we call giant viruses. And so we have viruses that actually have genomes that are bigger than some bacterial genomes, which is pretty surprising because we always think of viruses as having just little strands of RNA or DNA, but it turns out they can actually be pretty complicated as well. So does that help?
So 2 more questions. How new is this wastewater treatment system in this form?
Yep, so activated sludge has been around since the 1910s. I think 1913, and a big book came out in 2013 called 100 Years of Activated Sludge. You might be able to find it at the library. But yeah, it's been around for a little over 100 years. And we've played with different variations of it, but it's still the most widely used way for biological treatment of wastewater.
Great. So 2 questions related, and then we'll wait for more questions after. Cool. Do organisms have acidity, and do organisms contain salt, vitamin D, or numerous other live needed things?
Yeah, that's a great question. And so the microbes generally have certain pH ranges that they can sort of tolerate. And so you have microbes that can live at very, very low pH and very, very high pH, but they're probably different microbes. In wastewater treatment, we're primarily around neutral pH. So, you know, between 6.8 and say 7.5. And the microbes in this case are actually able to maintain the pH at the right levels themselves. But in other cases, when we're designing these bioprocesses, we have to take that into account and think about adding chemicals that might adjust the pH. So that's a good question about the pH. And then the second one, also a good one, about vitamins and salts. And so microbes, and again, I study microbes in the context of microbial communities. And so microbes interacting with each other and these really complex sort of ecosystems. And what we find is that not all microbes can make all their own vitamins, but they sort of split up the work. And so you'll have microbes present that can produce, say, vitamin D, it's a good example, and that or other vitamins or other growth factors, and they will actually share these with the rest of the microbial community. And so if you were to take a teaspoon of that wastewater, you'd count, well, if you any amount, you'd be able to count about 1,000 different species. And so the complexity in here, it's pretty robust, and these microbes are really good at sort of delegating different jobs to make the community sort of thrive. And so I hope that helps.
That's great. So you guys keep putting your questions in the Q&A box, but we're going to move along right now just to make sure we can get through the presentation today.
And then somebody did ask, after it's treated, where will it go? Sorry, I did just want to answer that. And so it does get discharged back into the environment. And so usually a lake or a river. Thanks for asking. All right, so how do we use microbes to make energy? So I have a question for you, and I'm curious again, if you know, and if you don't know, that's fine. But do you have in your house, do you know if your heating comes from propane, natural gas, or electricity? Propane. So I see people starting to answer. Propane.
And you may not know because you don't pay the bills.
Yep. Natural gas, wood and propane. There we go. Natural gas would prefer geothermal. I don't know. Cool. Natural gas. All right. And so in the last part of this talk, what I want to talk about is how we use microbes to make energy. And if I can get the slides. to the right place. So why would we want to make energy from waste? And I am curious to hear your answers this time around. And so if we can do it, why would we want to? Any good reasons you think that we could take waste and make energy from it? Laws of conservation of matter energy. It's cheaper. All right, and so one of the advantages is, I mean, there's really two advantages if you think about it. And so if we can take waste and make energy, it gives us a renewable source. And so if we can take a waste stream like dairy manure, which I'll give you an example of in a second, we can use that to capture the environment, and that gives us a feedstock that, unlike some other feedstocks for energy production, is renewable on human timescales. And so that's the main goal, is if we can use waste, it will help us reduce the amount of fossil fuels that we need to produce energy. So one way we do this as environmental engineers is using microbes, of course. And so what I've shown here, and these big white tanks, and they look really futuristic, but these are called anaerobic digesters. And we use anaerobic digesters to take a whole wide variety of waste, organic waste, and convert them into methane primarily. So we make what we call biogas, which is a mix of methane and CO2. And sometimes we can convert this into either combust it directly and generate electricity, or we can clean it up by removing a lot of the CO2 that's present and use that as natural gas directly. And so these tanks here, and this was the picture that I included that Lauren showed earlier, this is an anaerobic digestion facility at Goodrich Farm in Salisbury, Vermont. So I don't know if we have anybody down near there. But this facility was just opened, and they're actually still starting it up. But what they do at this facility is they take cow manure from the farm, and they take food waste from off-site, and they feed it into these tanks. And once in these tanks, microbes, and again, a community of microbes, convert that organic matter into biogas. And at this site, they're actually going to take the biogas, remove the CO2 using gas scrubbers, and Middlebury College is actually going to be buying that gas to heat their facilities. And so again, they're just starting up this facility, and my lab is working with them a little bit to sort of monitor the microbial communities during their startup phase. And so this, again, you can read about this. It's been in the news. This is a clip from the Vermont Digger. And so you can go read about these digesters. I love when anaerobic digesters get in the news. And the way that the anaerobic digestion works, it's actually pretty complex, but I want to just give you a sense for what happens, because in wastewater treatment, a lot of times microbes are kind of doing their own thing and sort of going after different feedstocks and living in harmony, but not necessarily sort of funneling food to each other. Whereas in anaerobic digestion, we start with these really complex feedstocks like complex carbohydrates, proteins, nucleic acids, lipids. And the first thing that happens is there's microbes that will excrete enzymes to hydrolyze these complex organics. And then what we end up with are these simpler organics that more of the microbes can eat. And through these processes, we end up with a couple of key intermediates namely acetate, CO2, and hydrogen, that get consumed by methanogens. And methanogens are the first archaea that we're going to be talking about. So these are archaea, and the rest of this community is primarily bacteria. And so the bacteria sort of initiate this whole process by breaking down complex things, funneling them into a couple of good food sources for methane to actually be produced. And so this is what the food web for anaerobic digestion looks like. And I wanted to just show this quick diagram to make a couple of points. And so this is a process flow diagram for the facility at Goodrich Farms. And this anaerobic digestion tanks are where the microbes are. And there's also microbes that are in this hydrolyzer tank that are actually initiating the breakdown of the complex materials. And since somebody asked a question about pH, the major difference between these two tanks with microbes in them is actually the pH, with the hydrolyzer being about a pH 5, and then the anaerobic digesters being about pH 7. But there's a whole bunch of supporting processes that go around anaerobic digestion. And so the key, one of the keys I want to show you, again, I mentioned that the biogas gets collected and it goes to a gas cleaning system. And so let's see if I can do this. Hopefully you can see that more or less. And so the gas gets cleaned up in a gas cleaning system. The other thing that happens is with the liquid residual, we can take that and we can actually remove the phosphorus and the nitrogen into separate products. And so what this allows us to do is, it gives the farmer more choices in terms of fertilizing their fields. And so initially, if they were just going to spread manure, you'd get the nitrogen and phosphorus combined as part of the manure. And what this allows us to do then is we can apply nitrogen and phosphorus separately. And this is a big deal because in Vermont, we have a lot of areas that are saturated with phosphorus. And phosphorus is a major contributor to water pollution in Lake Champlain. And so what we can do with these supporting processes is actually break up the nutrients into two separate streams. And so not only is it good for the environment in that we're making, producing renewable energy from a renewable feedstock, in this case, manure and food waste, but we're also better able to manage water quality impacts by separating these fertilizers into two different types. And so some take homes about anaerobic digestion. So microbes in anaerobic digestion, they primarily break down large complex molecules, and they use fermentation in this case. So by fermentation, we mean that they don't breathe in sort of the classical sense of the word. And at the end of the day, they're shuttling all of this organic matter into methane and CO2. And in like activated sludge, they're living microbes, and they're going to grow additional cells as they go through this process. But in this case, they're growing a lot fewer cells than an activated sludge. And so fermentation, you've probably heard about. And so fermentation is something that we use to make a variety of different food products. And in a lot of ways, those processes are similar to what we see in anaerobic digesters. So, all right. So that has, that's the third thing I wanted to talk about. Before we move on, I just wanted to let you all know that, again, I work at the University of Vermont, and our students, so me and my students are working on a variety of different projects related to using microbial communities to clean up and protect the environment. So we do a lot of work with anaerobic digestion. We do a lot of work with wastewater treatment. We also try and study microbes in sort of a non-engineered environment and in a natural soil environment, for instance. And at the end of the day, we're really interested in finding new ways to use microbes to help us protect the environment. And so with that, let's see. I just want to say thank you. And I'm happy to take any and all questions that you have right now.
Yeah, so we do have a couple of questions in the Q&A already, but before we go to that, and while you guys are thinking of questions you might have for Matt, I'm going to throw up just a quick poll. We always like to know what you thought of today's cafe, and more importantly, how much you learned. So it's a rating scale of 1 to 5, 5 being excellent. So I put the poll up. If you all could just take a minute to answer the poll, then we'll get to the questions, and be thinking of questions that you want to put into the Q&A box. So we'll just give it till I see most people having answered the poll. And then again, you can put your questions in the Q&A box. Yeah, I don't know why you guys can never see the poll. So you guys can put if you can't see the poll, put your choice, your answer in the chat box, and I'll make sure to add it. So I'm going to give about 10 more seconds. If a couple more of you could respond to the poll or put your answers in the chat box, that would be great. And then again, any questions in the Q&A box. All right, I'm going to close the Oh, thank you. One more person just got in there. I'm going to close the poll. And let's go to the questions. So first question, is methane bad for the environment?
That's a good question. And so methane is a very potent greenhouse gas. And so we need to be very careful if we are making it, that we capture it to the fullest extent possible. And so as engineers, if we're looking at an anaerobic digestion system, we have a pretty contained small system where we can generate this gas and collect it, and we capture the vast majority of it. And then we combust it and convert it to CO2, which then goes into the environment, right? But it's in terms of greenhouse gas potential, it's a lot better to have CO2 than methane going into the environment. And so methane, I can't remember the number off the top of my head, it's like 27 times as potent as CO2 is a greenhouse gas. I will say though, the nice thing about using something like anaerobic digestion, if, in Vermont, we can't do this anymore, but if you were to send your food waste to a landfill, it becomes a lot easier for the same sort of microbial processes are going to happen, but in a much less controlled environment. And so in those cases, we do see a lot of methane getting released to the environment, which is not ideal.
Yeah. What's the most important organism?
The most important organism. I think most people in anaerobic digestion would say that it's the methanogens. And the methanogens are the most important, not just because they produce the methane that we're all interested in, but they also sort of control, by consuming these products that are made, they help sort of drive how quickly these sort of processes can happen. And so methanogens, the archaea, are, if I had to pick one, it's not going to happen if you take any of those groups out, but if I had to pick one, I guess I would pick methanogens.
How effective is the process compared to other green energy sources?
Yeah, and so if you're thinking about solar, hydro powers, it on a footprint basis, which is one way to look at it, it's a very sort of compact way to produce energy. The other sort of service that it does for us too is it gives us some place to get rid of our waste and to treat our waste. But in terms of overall efficiency, if we can get all our power from solar when it's sunny, that's, I think, a great plan. It's hard to, I think, compare directly to something like a hydropower system, but I hope that helps.
In the chat, Nicholas is asking, are you trying to come up with a way to convert CO2 like Elon Musk has put out there?
Right. Yeah, so Elon is offering $100 million for the best CO2 capture technology.
Oh, better get on it.
And so I do work on CO2 capture a bit, and I also work on generating hydrogen. And I look at sort of combining those two molecules in ways that can benefit society, essentially, making a whole variety of chemicals. And so I do. I primarily look at capturing CO2 using microbes. So microbes that photosynthesize use CO2 as a food source, as a carbon source, and they use sunlight as an energy source. And so if we can mimic, and again, even if we took this outside of a microbial cell and mimic some of the enzymatic activity that we see, we can potentially harness some of that to fix more CO2 using sunlight in sort of a similar way that solar energy uses sunlight.
So our final question, KL is wondering if, I'm sorry, another question just came in, so it's not our final. If you would share your slides, can they email you if they would like to have a copy of the slides?
Yeah, that's definitely.
Okay. So you have Dr. Scarborough's email, I believe.
Yep,
his email's right there. So you can do that. And then, so this is complete this sentence. Your favorite subject in organisms is?
My favorite subject in organisms is, oh, that's a tough one. I'll pick transcriptomics. So I'm very interested in how organisms sort of decide which genes they're going to express and sort of what job they're going to do and what sort of environmental things trigger those changes in a microbe. So that was, I hope that is sufficient, but I like that question a lot.
Oh, we did get, yes, the question is your email the one on the screen? Yes, yeah. The mscarborough@uvm.edu is how you can reach Dr. Scarborough. And the www Emerge Lab is his lab on campus, so you can learn more about the work that they do. So I would highly encourage you all to go to the website just to learn more about his lab's work at UVM. And then reach out to him if you have any additional questions. Oh, we got more coming in here. Oh, no, that's just a thanks.
You're welcome.
So let's all just thank Dr. Scarborough for sharing his knowledge with us today and shedding light on this fascinating work of, taking something that is traditionally a waste product and finding value in it and helping the environment all the while. So you all know we have science cafes every Wednesday. I hope to see you all back here again next Wednesday. And yeah, thank you all for writing in the chat, thanking Dr. Scarborough, and we'll see you all in a week, hopefully.